which I think is what she's going to talk about today. So thanks so much for coming.
Thanks, Christina, for that introduction, and thanks to the organizers for inviting me to this conference. I'm going to take off my transcription epigenetic regulation hat and today tell you some of the work we're doing on cancer immunology, which is like RNA-based at least. So as we know, cancer immunotherapy has brought paradigm shifts to cancer treatment, and this year, Alison Hanjo got the Nobel Prize for developing immunotherapy. When we started exploration on this area 4 years ago, we ideally wanted to have patient tumors before, after immunotherapy treatment, or at least before treatment, but have their response to checkpoint inhibitor information. But 4 years ago, these type of data are very, very limited. So we thought maybe we'll just take these existing tumor profiling cohorts, and even though they are not related to immunotherapy and we only have pretreatment data, maybe we can still learn something about immunotherapy or microenvironment. So among the different profiling techniques, we focus on gene expression, because nowadays RNA-seq experiment has become so inexpensive. Like oncologists can isolate tumors, send to a CRO, they can do RNA extraction, library preparation, and sequencing together for about $200–300 a sample, right? So we believe there will be a lot more data coming in the next few years. And so the three questions that we want to ask is, how do the tumor-infiltrating immune cells influence patient survival? And how do immune repertoires inform us about the tumor immunity? And how to predict the patient response to immune checkpoint inhibitors? And I will present three algorithms we developed, the 3 Ts. So the first algorithm was to see whether tumor RNA data contain information about immune cell infiltration. The idea is we developed this algorithm first to filter the genes that we believe are uniquely expressed in the, so we select those genes that are uniquely expressed in the immune cells, and then for the expression of these genes in the tumor, we believe this is kind of a different immune cells mixed at different levels. And so we use the deconvolution algorithm to estimate the amount of immune cells in each tumor. So the goal is not to compare whether the tumor has more B cells or T cells within the same tumor. Instead, we're more interested in comparing for different tumors, which one has more CD4 T cell. For the same cell type across different tumors, we can have a good comparison. There are different algorithm developed, and there's a very good review of this from this review paper. But with this deconvolution across TCGA tumors, we could then examine how the different immune cell composition influence patient survival. In fact, one of the big effects is age and the tumor stage, but if we factor those out, we can see that in general, macrophage are worse for, bad for patient survival. So these red blocks are, which means that they are positive risk for patient death. Whereas CD8 T cells, you can see the blue means they are actually good for the patient. There are some cell types like B cells in this case, you can see it's quite inconsistent. We'll show you why that's the case. So with this, we decided to develop also a web server. And so if users input the gene they are interested in, the cancer type, the server can show you how the expression of this gene influence immune infiltration, how the mutation and copy number differences influence immune infiltration, and how gene expression and immune infiltration influence patient survival. We also have a estimation function, so you can upload your expression level to estimate the immune cell infiltration in the tumors. When we analyzed the data, we found this one interesting case. So in addition to PD-1 and the CTLA-4, which are expressed on T cells as an indication of T cell dysfunction, TIM3 is another important inhibitory checkpoint regulator that's expressed on the T cells. And so we believe the tumors that have high TIM3 should also have T cell infiltration. However, TIM3 is the highest expressed in kidney cancers, but in kidney cancer, we see some unusual cases where the TIM3 expression is super high, but these tumors were not having that high CD8 T cell level. And so we asked around the local collaborators. Turns out one of our pathologists specializing in kidney cancer, submitted some of the TCGA kidney samples to NCI, and she still has the tissue slides available. So she helped us stain the tissues. It turns out, so these are the normal ones. So you have low TIM3, low CD8. This is also normal. You have high TIM3 and high CD8. But this is the group that's kind of unusual. They have high TIM3 but low CD8 infiltration. Turns out that TIM3 is expressed not only in the T cells but also in the cancer cells. Right now there are TIM3 therapeutic antibodies under development, which hasn't been going through clinical trial yet. So we don't know the effect of this in clinical patient response, but we suspect that trials looking at this need to separate those patients where the TIM3 are only expressed in the T cells versus those where TIM3 are expressed in both CD8 and cancer cells. Yeah, so the first part is we developed this timer algorithm to estimate the immune cell components in tumors, and we also find that TIM3 expression can happen both in T cells and cancer cells in kidney cancer. The second part is we want to see whether we can use the tumor RNA-seq to estimate immune repertoire information. So we know that our immune system is able to develop a very diverse repertoire to recognize different antigens, including tumor antigens. This is through a process called VDJ recombination. So for example, our T cell receptor is going through this VDJ recombinations, by randomly selecting 1V, 1D, and 1J for the combination, and this junction creates a hypervariable sequence called CDR3, which is on the T cell receptor to recognize the tumor antigen or the viral or bacterial antigen. There are commercial solutions available for immune repertoire profiling, but these are kind of expensive. Also, if you only have limited amount of tumor sample, they have already gone through whole exome sequencing and RNA-seq, there will be very little sample available to do immune repertoire profiling. So we thought maybe we should just get it directly from tumor RNA-seq, because the RNA includes the expression coming from the T cells or B cells. Indeed, if we try to map the reads back to the genome, we can see there are some reads that are mapped to the V region. This is because, so this VDJ junction, the V is a pretty big fragment. So in TCGA data, when you have a 50 base pair reads, very often the whole read can be mapped to the V region. However, mapping the very short D region and J region is difficult because they are short, and also the junction, this yellow area, they create a lot of mutations and indels, which are not in the reference genome. In fact, when you do RNA-seq of tumor, the read mapping step will throw away most of the reads that are mapped to the VDJ junction sequence. So we developed this de novo assembly algorithm called TRUST, and from there we assembled 3 million TCR CDR3 sequences from the unmappable RNA-seq reads. When we analyze this across the 10,000 TCGA tumors, so this is all the TCGA tumors. The y-axis is what we call clonotypes per kilo reads mapped to the TCR region. It's a proxy for TCR diversity, and you can see here, in general, the tumors on the right side, these are the tumors with higher mutation load, which means they have more mutations, and also they have higher T cell diversity, suggesting that you will need more type of different T cells to recognize a diverse mutation in the tumors. In addition to the T cell going through VDJ recombination, our B cells also generate a diverse repertoire. And so the hypervariable sequence is responsible for antigen recognition, and the constant region is responsible for eliciting downstream different immune responses. Just from mapping the raw reads into the genome, we know that there are 10 times more BCR reads than the TCR reads, but there are some later complications, so we have to revise the TRUST algorithm. But from the revised algorithm, we were able to identify 30 million BCR, basically antibody sequences from the unmappable RNA-seq reads in TCGA. And so within the TCGA, we see about 400,000 clusters like this, So it's from the same patient. You can see the CDR3 sequences. They are highly, highly similar, except for occasional locations with mutation. It's unlikely that these are coming from completely different B cells that happen to recognize the same or the same antigen. More likely, what's happening is that there is one parental clone which has some recognition to the antigen. Then, as the B cell is going through clonal expansion, there's an enzyme AID which is creating additional mutations on the CDR3 sequence to gradually evolve an antibody with better and better specificity against the antigen. So that the fact that we see these type of clusters in the tumor is an indication that the B cells are recognizing the tumor antigen for clonal expansion. And if we look at the overall BCR antibody sequence distribution, you can see most of the antibody we see are between 8 amino acid to 22 amino acid long. And we know that the somatic hypermutation that we call is not random or not from sequencing error or PCR amplification bias, because we see this strong 3-base pair periodicity, because as the antibody is doing the mutation, the third base of the codon is not under selection, but the first 2 bases are under strong selection. This means that during the antibody evolution, the maturation process, there is strong selection in the tumor. And if we look at the somatic hypermutation patterns, we see more somatic hypermutation in IgG type of antibody, which agree with the current understanding of somatic hypermutation. In addition, we see that tumors with higher AID expression also harbor higher somatic hypermutation. So when the tumor actually see the the B cells recognize the antigen, in addition to the somatic hypermutation, the constant region of this antibody also gets shifted. And so initially the naive B cell is IgM, and then it's IgD, then IgG, and IgA. And different kind of Ig constant region will elicit different immune response. Since most of the TCGA data are paired and RNA-seq reads, very often from one end we can map the VDJ, and from the other end we will know the constant region. And so when we map the constant region of the tumors, we can see this, we do have across all TCGA, but you can see for breast cancer, maybe because it's a mammary gland, it secretes proteins, and so we see more IgA antibodies, whereas in melanoma, overall, we see more IgG antibodies. And sometimes we also see cases where the VDJ sequence is almost the same or in the same cluster, but the constant regions can have 2 different classes. That is the antibody going through active class switch, shifting the antibody in during the tumor evolution, like the B cell clonal expansion. And so the fact that we see this kind of enrichment of IgG class switch, in this case IgG1 to IgG3 class switch, is another indication that in the tumors, B cells are recognizing the tumor antigen. You probably remember from the timer part that B cells, we don't know whether it's good or bad for the tumor. Indeed, we look at many different publications, depending on the cancer type, depending on the cohort. Some people say B cells are good, and some people say it's bad. As a result, there is this review paper from Shiv Pillai's group saying that, there's a love-hate relationship. Indeed, it's kind of unclear. But if we look at the TCGA tumors that have at least 10 normal samples, we can see here, overall, if you just look at the B cell infiltration, indeed, in different cohorts, they are inconsistent. However, if we look at the B cell CDR3 diversity, you can see here, most of the tumors have a lower diversity. It's because of when you have clonal expansion, when it's more clonal, it's less diverse for the B cells in the tumor. In addition, we see a high enrichment of the IgG class switch, especially the IgG3 to IgG1 class switch. And so you can see this is definitely an enrichment in the tumor compared to the normal. It would mean that it's not the total amount of B cells in the tumor, but those that go through clonal expansion and Ig class switch that are important in the tumor. So in terms of the B cell response, the most important kind of attack is through something called ADCC, in which case, the cancer is expressing antigen. When it's recognized by the antibody, it's able to recruit natural killer cells through this Fc gamma receptor, which is also called CD16A. So we look at across TCGA tumors to see which of these tumors have their different constant regions are correlated with CD16A. So you can see, in general, if the tumor has more IgG1 or IgG3 antibodies, so this one is IgG1, 3 together, you can see the abundance of the IgG1, IgG3 are highly correlated with CD16A or natural killer cell activity, whereas for IgG4 or IgA antibodies, they are in general negatively correlated with natural killer cell activity. For example, right now in pharmaceutical companies, there is a PD-1 antibody and also PD-L1 antibody. You might think that they work similarly, but from this analysis, we would know that NK activity is going to kill the target cell. Therefore, if you want to have like a PD-1 antibody, you want to just block the PD-1, PD-L1 interaction, but you don't want to kill your own T cells. Therefore, PD-1 antibody should have an IgG4 constant region, but PD-L1 is targeting the cancer cells. In addition to blocking, you want the NK activity, and so there you want IgG1 or IgG3 antibody. So the constant regions, depending on your target, you want to use a different constant region. So when we see this in the tumors, so basically when the natural killer cell is being recruited to the cancer cell, there's a receptor called NKG2D. The way it recognizes this as cancer cell is because the cancer cell have, or actually our cells have a MICA expression in here. And so once the natural killer cells see the MICA, they will elicit the attack. Interestingly, tumor cell, there are 20% MICA amplification in the tumors, and we're like, why would the receptor recognize a cancer? And the cancer will still overexpress this to have better recognition. Interestingly, when MICA is amplified, it can be cleaved to form a soluble MICA. And the soluble MICA can internalize NKG2D, kind of like a shield to push the sword inside the NK cells. And so in some sense, MICA amplification and NKG2D is like a PD-L1, PD-1 pair between cancer cell and NK cell. And so we see that the MICA amplification in the tumors are actually associated with worse clinical outcome of the patient, suggesting that the patient that the B cell attack is in effect, and the patient tumor is evading the B cell or NK cell attack. And when we look at other clinical effect, it's kind of interesting. We see that overall, even though MICA amplification is bad for the patient, but if this patient have more IgG1, IgG3 antibodies doing the IgG Class Switch, their patient survival seems to be even better. We are asking ourselves, what is going on here? It took us a long time to realize in the tumors, the B cells don't just recognize mutation. They also recognize super amplified regions. For example, when the tumor has HER2 amplification, there are autoantibodies just recognizing the amplified HER2, and they have been shown to be better for the patient survival. And so we suspect that in these cases where MICA is amplified, there are autoantibodies specifically targeting those soluble MICA to kind of block their effect for NK. In fact, one of our collaborators, Kai Wuki-Fennig, was able to identify a therapeutic antibody directly from the MICA amplified patient, and he's developing this into a therapeutic immunotherapy. And so we believe there are actually additional useful antibodies in these 30 million antibodies that we identified. So, in summary of the second part, our algorithm trust is able to infer the tumor infiltrating TCR and the BCR repertoires, and can review that indeed in the tumors that there is B-cell mediated immunity, and the tumor is even amplifying MICA to evade the B-cell and NK cell attack. In fact, this algorithm, you can use it to analyze any kind of a tissue. We are also looking at AML samples or ALL samples. You can still look at the immune microenvironment in every tissue type of experiment. And the third part is we try to see whether we can predict the immunotherapy response from patients before they are treated with checkpoint inhibitors. And so here we get insights from a review paper from Thomas Gayaski, where he proposed two broad categories of tumor immune evasion. The first type is the T cell is able to get into the immune cells, but it becomes too exhausted or too dysfunctional. The second type is the T cell, for some reason, cannot infiltrate the tumor, which there are T cell exclusion things preventing the T cell from going in. And so we want to see whether in the gene expression data we can model these two types of situations. To model T cell dysfunction, we look at, again, these treatment-naive samples, not immunotherapy-related cases. For example, in TCGA melanoma cases, we see overall, if patients have high cytotoxic CD8 T cell level, the patient do survive better. However, this could be dependent on the expression of some genes, like TGF-beta1. This is a very well-known cytokine signal for a suppressive microenvironment. You can see when TGF-beta expression is high, even though there are T cells in the tumor, they don't really do much. They get too kind of drunk or exhausted. It's only when the T cell TGF-beta level is low, T cell can have its better function to protect the patient. And of course, TGF-beta is a well-known gene already. We want to develop a method to unbiasedly look at every gene to see how they interact with the level of CD8 T cells to influence patient survival. So we develop a interaction regression approach, test every gene to see whether when the gene expression is high or low, does that change the survival curve? We look at hundreds of cohorts to see which cohorts give us good enough signal, and there are 5 cohorts that have enough genes that meets our FDR cutoff. But you can see here there are 5 cohorts. Most of the top genes are quite consistent, and if we average their profile, we look at their agreement with previous mouse studies of T cell dysfunction, this average profile seems to do even better. And so we developed this signature for T cell dysfunction. It's a genome-wide signature. Every gene has a score, but most of the genes are driven by this really high positive score or very low negative score genes. When you have a gene expression profile, we correlate this expression with the signature. It can tell us how dysfunctional or drunk the T cells are in the tumors. And so here, because we only get a correlation, then we don't have to worry about multiple hypothesis testing or noise from individual genes. The next part, we try to model T cell exclusion. And so here, again, we get the insights from Thomas Gayaski. He mentioned many different cell types that might prevent the T cell from infiltrating the tumors. We just try to extract those expression signatures from GEO. And out of the different cell types, we found actually 3 suppressive cell types are really, really predictive. They are myeloid-derived suppressor cell, tumor-associated macrophage, this is M2 macrophage, and cancer-associated fibroblasts. Each of them is a genome-wide expression signature. And then we can evaluate, for example, again, this is TCGA melanoma tumors. The x-axis help us, the x-axis measures whether the tumor has a strong signature of each of these signatures. So the y-axis is the CD8 T cell level. So you can see each of these suppressive cell types give us a negative correlation. And if we average those two, we get an even stronger correlation, which means if the tumors have any of these 3 things, they will not have very high T cell infiltration. Indeed, we checked hundreds of solid tumor cohorts, and in all of them, this average signature have a negative correlation with T cell infiltration. And so then we derive this signature called like this exclusion signature, which basically, again, is a genome-wide signature. If you have another gene expression data, we can correlate this expression with this genome-wide signature to measure whether there are door blockers preventing the T cells from infiltrating the tumor. And so putting these two together, we developed this TIDE model, which evaluated both the T cell dysfunction in the immune hot tumors and the exclusions in the immune cold tumors to predict the patient response to immunocheckpoint inhibitors. So the algorithm works like this. If we have the tumor gene expression, first we ask, it is T cell high tumor or CD8 T cell low tumor. If it's kind of an immune hot tumor, we ask are the T cells dysfunctional or dysfunctional or exhausted by correlating the tumor expression with our dysfunctional signature. This is just one example of a CTLA-4 cohort in melanoma. You can see, so these are the immune hot tumors here. When they have high T cell dysfunction, they are more likely to be non-responders. Otherwise, they are more likely responders. But yeah, so these are dysfunctional. But if this tumor is an immune cold tumor, we correlate with the exclusion signature to ask whether there are door blockers preventing the T cell from going in. And you can see if there are, the patient almost never responds to immunotherapy. But otherwise, even if it's an immune cold tumor, maybe immunotherapy can still stimulate the T cells to infiltrate the tumor. So let's look at the TCGA melanoma samples. Each dot is one tumor. Red is a hot tumor, blue is a cold tumor. And the x-axis measures whether there are door blockers preventing T cells from going in, and the y-axis measures whether the T cells are too exhausted. Even though none of the patients were treated with immunotherapy, we can make some guesses. For example, these tumors, even if they are very, very hot tumors, their T cells are probably too drunk to really work, to be waken up by immunotherapy. And these tumors, not only they are very, very cold, they have door blockers preventing the T cell from infiltrating. Therefore, they are unlikely to respond to current single agent immunotherapy. But these middle patients, they are more likely responders with current either PD-1 or CTLA-4. So we look at this across a number of immuno-oncology cohorts here, compared to PD-L1, interferon gamma level, total mutation load, and there are more in here, CD8 T cell infiltration level, T cell clonality. There's a computational algorithm called IPS, and also somatic copy number variation, which has been reported to be important. You can see here, TIDE can outperform all of them. And this last cohort I want to mention, this is actually a nanostring cohort with about 800 genes, not genome-wide RNA-seq, and we still have predictive power. And in this cohort, in addition to melanoma, there is also lung, adenocarcinoma, and squamous, and there are several other smaller cohorts, smaller tumor types as well. In addition to predicting patient response, we also see that this algorithm can predict patient survival or progression-free survival, which means that before the patient were even treated with therapy, based on their tumor microenvironment, we can see this patient has better immune potential to be waken up by immunotherapy. And so we made a website. If you have a cohort, you can submit your tumor. We can tell you, whether it's a CTA high or low, whether the T cells are too drunk, or whether there are other cells preventing T cell infiltration. We have many. We continue to add to this, and also this query gene. You can see it later. If you enter a gene, it will tell you how it performs in those 5 cohort, as well as we have about close to 200 cohorts now, and also current CRISPR screen data and immunotherapy trial results to see whether this gene is important or not. We believe that in addition to having predicted just a biomarker, some of these genes might be important regulators of immune checkpoint inhibitors, which might be targeted for immunotherapy. For example, this is the CD274, this is PD-L1, that's the important target. TGF-beta, that's what we showed in the example, but we also want to show this gene, SERPINB9. Recently, there is a mouse study from Andy Min's lab where they look at a mouse treated with anti-CTLA-4. And when they compare the parental cell, that's kind of tumor that are respond to immunotherapy versus those that became resistant, looking at all the genes that are predicted as the high dysfunctional score genes, the top differential gene is this SERPINB9 gene in here. If we look at the published immuno-oncology trials compared to the responders and non-responders in here, you can see most of the non-responders have high SERPINB9 gene expression. Earlier this year, we were collaborating with Kai Wuki-Fenix's lab to look at cancer cells co-cultured with T cells to see if we knock out some genes in cancer cells, whether that influence the T cell ability to kill the cancer cells. In the original study, we found the 3 protein complex, PBRM1, ARID2, and BRD7. These 3 are members of the switch SNF PBAF complex. So we published this in the original study, and there was a companion science paper showing that in the kidney cancer clinical trials, patients who have the PBRM1 mutation are indeed more responsive to immune checkpoint inhibitors. But you can see here, SERPINB9 is another gene that's a really strong hit. And so we individually validated this gene. You can see when we knock out the SERPINB9 in the cancer cells, the T cells can kill the cancer cells better. And if we overexpress SERPINB9, then the T cells cannot kill the cancer cell anymore. So what is SERPINB9 gene? Normally, SERPINB9 is expressed in the T cells and NK cells because when the T cells detect the cancer cell or recognize the cancer cell, it will use porphyrin to punch a hole on the cancer cell and secrete a granzyme into the cancer cell to make the cancer cell die. But when the T cells and the NK cell have the SERPIN in its own cell, how does that prevent the T cells from dying themselves? They put these they put the granzyme in granules, but in case of granule leakage, they also express SERPIN to inhibit the granzyme activity. And so SERPINB9 is a gene that's kind of a self-protection mechanism for T cells and NK cells from the granzyme B damage. However, if we look at, this is a public resource called Protein Atlas. They stained thousands of different proteins in different tissues. We can see that SERPINB not only is expressed in T cells, in this case in melanoma cells, we can see it's usually very highly expressed in the cancer cell as well, which means that the cancer cell actually hijacked a T cell mechanism. It's like, oh, if you can use SERPIN to highly express to protect from granzyme B, then the cancer cell themselves can also highly express the SERPIN to prevent granzyme damage. So there is a paper from Tim Chan's group in 2016 showing that SERPINB3/4 mutation to be associated with better outcome in melanoma patients with CTLA-4 treatment. So they are in the same family, but out of all the SERPIN family, SERPINB9 is the best inhibitor against granzyme B. And recently, there is also a cell paper from Aviv Regev's lab for immunotherapy treated patients. And again, we see those patients who don't respond to immunotherapy, their cancer cells are overexpressing SERPINB9. And so in summary, so we have developed this tied model to study T cell dysfunction and exclusion to predict the response to checkpoint inhibitors. We also identify SERPINB9 as a cancer cell intrinsic mechanism. It hijacked the T cell mechanism to resist T cell mediated attack. And we also have this website. I hope that this can show you that the tumor RNA-seq, even if they are not immunotherapy related, even if the patient hasn't been treated with anything yet, is still a very cost-effective approach to give us insight on the tumor microenvironment and tumor immunity. And so some of the ongoing work that we are doing, so right now we have about 30,000 different tumor samples. Even though they are not immunotherapy related, we can still use them to learn the immune features and use a deep learning model to kind of look at the interaction between the different immune signatures, to look at their survival, mutation, other kind of outcome. And then we use these features learned, so we keep all the parameters in the early on training to then look at immunotherapy treatment where we only train the last layer of immunotherapy. It's kind of a transfer learning approach to model the immunotherapy response, and we see really good results. Also, we are trying to use a deep learning approach to see if we have the repertoire of the immune BCR, so in this case, antibody sequence. We want to first see whether we can see the amino acid, do they share different properties, and then we look at the K-mers within the immune repertoire. We also then model the antibody isotype, the different constant regions, and then for each repertoire, we do a collection of different K-mers. At the end, we try to ask, does that predict, at least we want to see, does that predict based on the repertoire that this is lung cancer and that's a melanoma and that's breast cancer? And we actually get really good results. And then we try to see whether we can use this approach to predict the expression of some genes, and maybe these genes are potential antigens. And we also have prediction. And so right now we are in the process of testing some antibodies we see in the tumors to see whether we can predict the correct antibody for the antigen. Then we can potentially develop therapeutic antibodies directly from our tumors. So of course, we're not limited just to look at tumor RNA-seq. Recently, we are involved in this CIDC Immuno-Oncology Biomarkers Network. So basically, NCI want to evaluate all their immuno-oncology trials and send them to profiling in these 4 profiling centers. And there's a layer of different assays, like from genomics to imaging, proteomics, some epigenetics, depending on the availability of the samples. And we are the data center. And so our goal is to integrate all of these different profiling and collaborate with the profiling centers and the trial centers to identify biomarkers for optimizing immunotherapy strategies in cancer. And so when this grant was funded last fall, 11 pharmaceutical companies decided to join forces, and actually by now there are 12 companies. They devoted resources, and they also want to kind of share our insights, and also they want to share some data. The goal is to really accelerate the development for new immunotherapy treatment. So I won't say immunotherapy will really completely cure cancer. I think chemotherapy and targeted therapy are still probably very, very important because they specifically kind of kill the cancer cells. Immunotherapy is kind of different because it kind of boosts your own immune system and indirectly kill the cancer cells. I have long been working on epigenetics. Even though these drugs haven't been as promising in clinical trials, very often when we do CRISPR screens of drug treatment, we see epigenetics genes showing up, suggesting that maybe instead of as a single agent, it can be used as a combination therapy with other drugs. And so you can see these 3 types of therapies are very complementary. So epigenetics probably a modulating the tumor microenvironment system balance, right? So in order to really best combine these 3 different type of therapies, we believe that the tumor profiling, it doesn't matter whether it's a bulk RNA, DNA, immune repertoire, or single cell profile, epigenetic profile, is still very, very informative. CRISPR screening is also very powerful approach. It's a functional screening, giving us insights on if we get rid of a gene or inhibit a gene, would that give us a real functional phenotype? And of course, these generate a lot of high throughput data, and we are developing computational models with the goal of really combining these 3 therapies together to cure cancer. Maybe not in 2020, but hopefully in the next 30 years. So I want to thank members in my lab. So Boli developed the most of the timer and the trust algorithm, which now has been taken up by Sherlock and Jian. And Peng was the developer for the TIDE algorithm, and the Stan is helping us with validation, and Taiwan developed the timer website. And we also collaborate with a lot of our colleagues. So Kai Wuki-Fennig is our main CRISPR screen T-cell dysfunction and co-culture screens. Miles Brown has been my long-term collaborator, and Jun Liu is our statistical colleague for the modeling, and Sabina Signoretti helped us validate the Team 3 kidney cancer samples. And I want to thank our funding agency, and we are recruiting. By the way, we're still working on epigenetic, continue to develop Systrom, and we are now looking at the single-cell RNA-seq and ChIP-seq and Systrom to see whether we can look at immune cell gene activation in the single-cell data. So I would love to work with talented people here and collaborate. Thank you very much.
Okay, we have time for some questions. I can start off with a question as people line up. So I think it's really impressive what you can do with bulk RNA-seq, but I wanted you just to speak to kind of caveats of bulk versus kind of more targeted analysis. and specifically, we aren't doing this, but collaborators are looking at single-cell RNA-seq versus deconvolution to analyze immune cell populations, and it's not so consistent, right? And so maybe you could just talk about limitations.
Yeah, so I think single-cell data is going to be really, really important, right? So we are in the process of looking into that data. However, in terms of having from the single-cell suspension to making a library for sequencing, apparently different immune cells and tumor cells have a different ability to get there. And so what you get from single-cell may or may not really recapitulate the actual tumor content. And also to train an algorithm, I don't think you can just use one tumor. You have to use multiple tumors in every tumor type to get a better signature. And so maybe for that, flow cytometry, CyTOF are still very, very important technologies as well. I think immune cell infiltration is definitely a topic. And for that, I also want to applaud the group that developed CyberSword. I know initially we have some kind of back and forth. They continue to update this algorithm, and it's actually getting really good. Yeah.
I'll just hold it. Thanks so much for a great and very expansive talk. I had a question about your TGF-beta story, looking at TGF-beta expression as a marker of resistance to immunotherapy. Now, I'm not an expert, but I do know a little bit that one of the ways I think that TGF-beta is thought to act as an immunosuppressor is by encouraging the development of CD4 regulatory cells to downregulate or turn off the CD8 cells. Did you see that in your timer results, like a higher amount of predicted CD4 cells in the tumor microenvironment? Is that something you looked at?
So it's not necessarily the total level of CD4. It could be the activity of the CD4, the activity of the CD8. So in general, if you see the high TGF-beta, we see actually a lot of other things that are correlated that show that this whole tumor is like in a suppressive microenvironment. And that's why we want to use this deep learning model, because every paper is showing, this is predictive of immunotherapy, that is predictive of immunotherapy. Maybe they're all correlated. And we're hoping that the deep learning can automatically learn these associations.
Thanks.
Just have a question. Have you applied these algorithms on hematological malignancies, multiple myeloma or something like that? Or do you expect this will still work in this situation?
You mean for immunotherapy or for getting the trust immune repertoire information?
Yeah, so some of the algorithms that you presented today.
Yeah, so initially we just did a TCGA. Recently we are applying the whole tide to the AML samples, but we are trying to get the St. Jude has like 3,000 RNA-seq samples in all sorts of different pediatric cancer samples. We're trying to get that data so that we can run like tide across them. And right now for all the cohort that we have, it's on the TIDE website. You can see each gene, how that gene is related to the overall cancer risk, how does that influence the T cell dysfunction. We probably have some multiple myeloma, but not a big number of cohorts. But it's just availability of data. So if anybody have tumor cohorts, even with minimum amount of clinical annotation, doesn't have to be immunotherapy treated. We would love to look at that.
Okay, let's just talk afterward.
Okay, great.
So one question about the tumor purity problem. How do you think, like for the bulk RNA-seq data, if it's not pure like tumor, it's a combination of normal and tumor that will affect the signature for their prediction power?
This is for the timer algorithm, right?
Yeah.
So for timer, because we started this TCGA data, let me see, can I get the slides? Yeah, so for this algorithm, we only use the tumor purity to decide which genes are kind of highly expressed in the immune cells. So depending on the tumor cohort, we use a different set of anchor genes. But once you have the anchor genes to get the final immune infiltration level, we do not need the purity information anymore. Obviously, purity will tell you, like so for example, initially you might see a lot of immune cells to be positively correlated simply because it's confounded by tumor purity. And so for a lot of this website analysis here, we conditioned on tumor purity so that you can take that factor out. You see, if knowing the tumor purity, are these two correlated or not?
I see.
Thank you for your very interesting talk. Can you go to the slide of the deep learning work that you've been working on? I think that's the first.
We're still doing this, so.
Yeah, but I'm kind of interested in the idea that because you demonstrate that in the first layer after the gene expression, you have immune features. So I'm interested in that. How can you extract that features? Probably like when you regularize the deep learning neural network and you get some weight in that layer. And also, I think the immune features in this layer is sort of like a linear combination between the gene expressions, or would that be possible if you push that to further layer, you can have some nonlinear combination between the gene expression, but just a thought?
Yeah, so first of all, this is still method under development. We just thought if we use all the genes, there will be too many parameters to train. So instead, if you just use like gene set enrichment to get signatures, you can have a much reduced feature size for training your deep learning. So the first part is not even deep learning. The first part is just like single sample gene set enrichment. Yeah.